Today we are going to learn about the phases of the microbial growth. When a few bacteria are inoculated into a liquid growth media and the population is counted at the intervals, it is possible to plot a bacteria growth curve that shows the growth of the cells over time. Okay. Here there are mainly 4 phases during the growth of the bacteria. That is, first phase is lag phase, second one is log phase, and third phase is stationary phase, and final one is death phase. Observe the diagram here. This is the bacteria growth curve simply. So these are the various phases of the growth of the bacteria. So this is the pattern of the bacteria growth. So observe here. This is the bacteria growth curve, which mainly shows the various phases of the microbial growth. Here time is taken on x-axis. On y-axis we will take the log of the number of bacteria. So we will get a pattern like this. The first phase is the lag phase. Here there is no increases in the number of bacteria here. And the second phase is log phase. During this phase, there is an exponential increases in the number of living bacteria cells is here. And the third phase is stationary phase. During this phase, the rate of the cell division and the death equal. And the fourth phase is the death phase. In this, the exponential decreases in the number of the living bacteria is seen here. The first phase is the lag phase. For a while, the number of cells changes very little because the cells do not immediately reproduce in a new media. So it takes some time that there will be some lag initially after the inoculation of the microbes. So this period of little or no cell division is called the lag phase. So here it can last for 1 hour to several days. During this time, the cells are dormant in nature and then the microbial population is undergoing a period of intense metabolic activities during this phase. During this phase, the microbes will build up the essential things needed for the growth of the microbes, like synthesis of enzymes, synthesis of various molecules which are needed for the growth of the bacteria. So this is about the lag phase. And the next phase is the log phase. Eventually the cells begin to divide and enter a period of growth or logarithmic increase called the log phase. This is also called as exponential growth phase. During this phase, the growth of the microbe is very high. Here the cell reproduction is most active during this period. The generation time reaches a constant minimum at this stage. What is generation time means? Time required for a cell to divide, that is its population to double, is called the generation time. So because the generation time is constant here, a logarithmic plot of the growth during the log phase is a straight line. The log phase is the time when cells are most active metabolically and is preferred for the industrial purposes. Here microorganisms are practically sensitive during the log phase to the adverse conditions. At the same time, radiations and many antimicrobial drugs, for example the antibiotic penicillin, exerts their effect by interfering with some important steps in the growth process and are therefore most helpful to cell during this phase. This is about the log phase. And the next phase is the stationary phase. If exponential growth continues unchecked, startlingly large number of cells could arise. For example, a single bacterium dividing every 20 minutes for only 24 hours can theoretically produce a population equivalent in weight to that of an 80,000 ton aircraft carrier. In reality, this does not happen. Eventually, the growth rate slows and the number of the microbial deaths balances the number of new cells and the population stabilizes. The metabolic activities of the individual surviving cells also slow at this stage. This period of equilibrium is called the stationary phase. What causes exponential growth to stop is not clear always, but the exhaustion of the nutrients, accumulation of the waste, and the harmful changes in the pH may all play a key role for the decreasing in the growth of the bacteria here. In specialized apparatus called chemostat, a population can be kept in the exponential growth phase identity by draining of spent media and adding fresh media. So this type of continuous culture is used in the industrial fermentation processes. So chemostat will be used there. This type of continuous cultures are maintained in the industrial fermentations by using the chemostats. So this is about the stationary phase. And the final phase is the death phase. The number of deaths eventually exceeds the number of the new cells formed and the population enters the death phase or the logarithmic decline phase. So this phase continues until the population is diminished to a tiny fraction of number of cells in the previous phase or the population die out entirely. Some species passes through the entire series of the phases in only a few days. So others retain some surviving cells almost identically. So this is about the death phase. So these are all the various phases of the growth of the microbes, that is lag phase, log phase, stationary phase and the death phase. Thanks for watching this video. Please subscribe to our channel for more videos.